Hello, my name is Naomi Karaluk, and I will be doing my biochemistry project on beta oxidation of fatty acids. So here I just have a picture of what that process looks like and what we will be getting into today to give you a little bit of an idea. So, an overview. What is beta oxidation of fatty acids? So, beta oxidation of fatty acids is the catabolic process where fatty acids are broken down in the body to produce energy in the form of ATP. And so what this means is from when we consume fats in our diet, how our body utilizes that and how it's converted into energy. So that's what we're going to be getting into. And it's a catabolic process because it is broken down bigger things into simpler things, which is the fatty acid into ATP. And in my diagram here, I have, you can see dietary fat just over there. So what happens when we consume dietary fat, which includes anything with oils in them, meats usually, eggs, cheese, all sorts of things in our diet, how that is broken down is it is stored as triglycerides in our body. So triglycerides are stored in our adipose tissue, in the adipocytes, and it can also be stored in our blood. And so these triglycerides, as you can see on the diagram, it is a glycerol molecule with 3 fatty acid chains attached. And so for beta oxidation, we need to have the fatty acids on their own. So this triglyceride is actually broken down in our body with the help of the enzyme lipase. So lipase is produced by the pancreas. And once this is broken down, we're going to get a free fatty acid, as you can see in my diagram. And there's an R group. So that R group is usually about a 14 to 16 carbon chain. And so when we go through beta oxidation, that free fatty acid is broken down by 2 carbons each cycle. So we want to completely break that fatty acid down, and so the beta oxidation cycle just continues until that fatty acid is completely broken. And for a 14 to 16 carbon chain fatty acid, it usually takes about 7 cycles of beta oxidation to completely break down a free fatty acid. So what are fats? So all types of natural fats, such as saturated and unsaturated fats, classify as a subgroup of lipids that are triglycerides, which we just talked about. So lipids are just defined as various organic molecules that are insoluble in water. So that's phospholipids, triglycerides, and steroids. We will just be covering triglycerides. But fats are very essential to our body's use. So they're essential to provide our bodies with energy, and we get that through beta oxidation, and they're very crucial for being used as energy stores in our body. So that's adipose tissue. We need these energy stores because it helps maintain healthy cell membranes, especially during starvation, prolonged fasting, low intensity exercise, and high intensity exercise when our body runs out of glucose and it needs to reach for our stores. And we reach for these stores to keep our cells healthy and maintained because that is crucial and our cells need to constantly be fed. Fats are also very important for vitamin absorption of fat-soluble vitamins. So these vitamins are vitamin A, vitamin D, vitamin K, and vitamin E. And these vitamins actually are useless without the help of fat. So if we ate these without fat, they would do nothing because they're fat-soluble. They're only absorbed with the help of fat. And these vitamins are crucial for our eyesight, our immunity, our bone health, and our blood health. So these are really important things, and we need to consume fat for our bodies. So why is this process called beta oxidation? So it's called beta oxidation because if you can see in this diagram I have there, that is a fatty acid chain, and that's just a 4-carbon fatty acid chain. But as you can see, the beta carbon of the fatty acid is the one that undergoes oxidation to the carboxyl group. So that makes pretty sense of why it's called beta oxidation of fatty acids. The theory of beta oxidation was actually formulated by a biochemist named George Franz in 1904. And this is when he studied biological degradation of fatty acids with his classical experiments. So this is when beta oxidation was fully discovered and formulated. So in order for beta oxidation to occur, we have to get that fatty acid into the mitochondria of the cell. But before that, we have to activate the fatty acid. So that fatty acid, once it's broken off the triglyceride, it travels through the bloodstream to then enter the cells and undergo beta oxidation. And this happens in most cells in the body for most tissues. Excluding the brain. The brain cannot use the energy from fatty acids. But as you can see over there, we have a prokaryotic cell, and I've circled the cytosol and I've pointed out the mitochondria. So this fatty acid has to enter into that cell, and it first enters through the cytosol. So the cytosol is just the aqueous component of the cytoplasm of the cell, so it's just the liquid and the surrounding areas. And so I'm just going to put my camera away so you can see my diagram. But that, as you can see, that free fatty acid has to enter the cytosol, and it does that with the help of fatty acid transport proteins. So these transport proteins help it enter into the cytoplasm, and once it's in the cytoplasm, the cytosol, the fatty acid has to be activated. So it's activated by being converted into acetyl-CoA. This happens when the enzyme acetyl-CoA synthase allows the CoA group to bind to the fatty acid chain, which activates it by converting it to acetyl-CoA. So as you can see in my diagram, the acetyl-CoA is in the cytosol, but then it has to eventually enter the mitochondria. But it has to go through a process in order to do that. But if it's a short-chain fatty acid, so smaller than 14 carbon chain, it can actually just diffuse freely across the membrane into the mitochondria. But for our most common fatty acid chains, it has to undergo a process of the carnitine shuttle. So as you can see, we have the cytosol of the cell, we have the acetyl-CoA in there, and in order to enter the mitochondria where it can undergo beta oxidation, it actually needs to be converted into acetylcarnitine. And it does this with the help of the enzyme carnitine palmitoyl transferase 1. And so what that does is just adds a carnitine group onto the acetyl-CoA, and then it can diffuse into the inner membrane space of the mitochondria. So as you can see in the middle there, the acetylcarnitine can then get into the matrix of the mitochondria with the help of the enzyme carnitine acetylcarnitine translocase. So then it can diffuse into the matrix. But we know our active form of that fatty acid is the acetyl-CoA. So what we do is the enzyme carnitine palmitoyl transferase 2 takes that carnitine away, converts it back to acetyl-CoA, just throws that carnitine back into the inner membrane space for further use. And now we have our acetyl-CoA in the mitochondria, which can undergo beta oxidation. But it's not always the case. If the fatty acid chain is too long, that's over 16 carbons, it must first undergo peroxisomal beta oxidation. So what this is in the peroxisomes of a eukaryotic cell, it must be processed and oxidized to break down a couple carbons before it can go through the carnitine shuttle. So the peroxisomes are just membrane-bound organelles found in the cytoplasm of eukaryotic cells. So as you can see there, they're just sitting in the cytoplasm, and that's what oxidizes the really long fatty acid chains. Through this process, it produces lots of water and heat, but then it is totally good to go into the carnitine shuttle to then enter the mitochondria. So in what conditions can beta oxidation take place? So in a fed state, the fatty acid beta oxidation is probably not going to happen because we're already fed. We don't need to get any more energy. But in a fasted state, when our body needs energy, that's when the catabolism is going to happen and the fatty acids are going to be broken down to form energy. So in a fed state, what's more likely going to happen is those fats, triglycerides, are going to be stored in our body. And this whole process is actually regulated by the hormones insulin and glucagon, which are produced in the liver. And it's regulated by these hormones because in a fed state, insulin is produced to lower blood sugar, which means that the process is not going to happen. But in a fasted state, glucagon is produced, which is going to regulate the process of the carnitine shuttle. So that's when beta oxidation takes place. So, actually getting into beta oxidation. So, once that acetyl-CoA is in the mitochondria, there are 4 steps to the beta oxidation. So, as you can see, in blue circle, I have dehydrogenation, hydration, oxidation, and thiolysis. So, there's 4 basic steps to this beta oxidation. So the very first step is called dehydrogenation, and I have it circled on the picture. So in the first step, we start off with our acetyl-CoA. So that acetyl-CoA uses the enzyme acetyl-CoA dehydrogenase to undergo a dehydration reaction and produce trans-acyl-CoA. So through this dehydrogenation reaction, the hydrogen atoms are removed from the beta and alpha carbons of the acetyl-CoA. They're actually donated to the cofactor FAD, which then produces FADH2. Those 2 hydrogens are added on. So that FADH2 actually goes on to the electron transport chain to produce ATP, and we are left with trans-acyl-CoA. So that trans-acyl-CoA can then undergo the second step, which is hydration. So during hydration, H2O is added to the trans-acyl-CoA and catalyzed by the enzyme acyl-CoA hydratase. So when this happens, this adds an OH group to the beta carbon of the trans-acyl-CoA to form hydroxysyl-CoA. So now we have hydroxysyl-CoA with a OH group added on. There is nothing produced from this step. Third step, oxidation. So now that we have our hydroxysyl-CoA can undergo oxidation. So what happens is hydroxysyl-CoA is catalyzed by the enzyme hydroxysyl-CoA dehydrogenase. It is also oxidized by the cofactor NAD+. And this removes the hydrogen from that OH group to form ketoacyl-CoA. So the hydrogens that are removed are then accepted by that NAD+ to form NADH. And NADH can then go onto the electron transport chain like the FADH in the first step, and then that's where ATP is produced. Now we are left here, I'll just show you that. We are left with our ketoacyl-CoA. So what happens with that is it is actually cleaved by a thiol group of CoASH and is then catalyzed by the enzyme thiolase. And this forms 1 molecule of acetyl-CoA and 1 molecule of acetyl-CoA. So as you can see in my diagram over there, I have those 2 molecules side by side as the 2 products of this reaction. And this is the last step of the beta oxidation. So what happens to these 2 products? So the acetyl-CoA is actually what we use for energy. So that one's going to go on to the Krebs cycle, also known as the citric acid cycle, and this is to produce ATP. But sometimes, in some cases that we'll get into, it can go on to the ketone bodies synthesis. But normally it's just going to go on to produce ATP. And then we also have our acyl-CoA. So that acyl-CoA is now 2 carbons shorter, and it's actually just going to keep continuing on that beta oxidation cycle until it is completely broken down, which we mentioned is going to be about 7 cycles. So 7 acetyl-CoA are going to be produced, and acyl-CoA will be completely broken down. Ketone bodies. So like I said, sometimes the acetyl-CoA can go on to ketone body synthesis. So this happens when your body's in a state of starvation. So the acetyl-CoA will undergo ketone body synthesis in the liver to produce ketone bodies to fuel the brain. So ketone bodies are just an alternative fuel that are formed when the body is really low energy supplies. So when there's low glucose, and this is just a quick fix to fuel our brain, because as soon as the brain is not fueled for long enough, it can cause sudden death or serious brain damage. So this is kind of a last resort that our body does. Our body will also produce ketone bodies overnight, when we're starvation, and when we're fasting. Okay, so the enzymes utilized during this beta oxidation. So we just went through the whole process of beta oxidation, and there's 4 enzymes that are used throughout that process. So those are acyl-CoA dehydrogenase, acyl-CoA hydratase, hydroxyacyl-CoA dehydrogenase, and thiolase. So these 4 enzymes seem to be individual, but they actually come together and form a trifunctional protein complex of alpha and beta subunits. So they're actually not individual enzymes, as though it might seem that way. They actually form a full complex together, and they're always together. Energy yield. So through that whole process of beta oxidation, as you can see over there, I have the 1 cycle of beta oxidation. So 1 cycle of beta oxidation is going to create 1 FADH2, 1 NADH, and 1 acetyl-CoA. So theoretically, FADH is going to produce 2 ATP, NADH is going to produce 3, and acetyl-CoA is going to produce 12. But this is theoretical because it is likely going to be a lot smaller due to energy loss in transport and not all the substrate being utilized in the reactions. So what the actual yield is going to be about 1.5 for FADH2, 2.5 for NADH, and 10 for acetyl-CoA. So the total theoretical yield would be approximately 17 for 1 cycle of beta oxidation, but the actual would be more approximately 14 ATP. So as you can see above, I put for 7 cycles. So in order to break down that entire fatty acid molecule, it's going to go through 7 cycles, right? So that's going to create 7 molecules of FADH2, which is going to equal 10.5 ATP molecules, 7 NADH, which will be 17.5 ATP, 8 acetyl-CoA, which will be 80 ATP, and that's going to make a total yield of about 108 ATPs. But we're going to take away 2 ATPs for the fatty acid activation, which takes some energy, and then we're going to end up with a total approximate yield of 106 ATP molecules that are generated from the breakdown of 1 fatty acid chain. So this is a lot of ATP, and actually fatty acids are the most abundant source of energy and provide twice as much energy as carbohydrates and protein. But as you can see, it's a large process for our body. So carbohydrates and proteins are actually, our body prefers them for energy over fats, but this is what happens after all the carbohydrates are broken down and all the proteins are broken down. So what is ATP anyways? So ATP is just energy used on a cellular level. So in every cellular organism, ATP is the form of energy. So ATP pretty much delivers energy within the cells of our bodies to maintain healthy cell function. So without ATP, our muscles, our nerves, our DNA synthesis would not work. We need our cells to maintain healthy and have energy to keep us functioning. So ATP just stands for adenine triphosphate, and I have the molecule just above so you can see what it looks like. So how does beta oxidation yield ATP? So as I said earlier, FADH2 and NADH go onto the electron transport chain, and the acetyl-CoA is going to go onto the Krebs cycle. This is how they make ATP. So what are those things? So the electron transfer chain is just a series of protein complexes that undergo redox reactions, creating an electrochemical gradient in the membrane within the mitochondria. And this is what leads to the creation of ATP. So I have a small diagram right below of what that electron transport chain looks like within the mitochondria. And the Krebs cycle, which is also known as the citric acid cycle or the TCA cycle, it's actually a part of the process of aerobic cell metabolism. So this is where molecules are broken down in the presence of oxygen into carbon dioxide and water to release chemical energy in the form of ATP. So I'll just show you quickly what that Krebs cycle looks like. That's where the acetyl-CoA goes. Disorders associated with beta oxidation. So genetic disorders of the beta oxidation pathway may cause metabolic diseases, and this makes sense. So if we have any genetic disorders that affect this process, it's going to be hard to get energy from fatty acids. We're not going to be able to do the full beta oxidation cycle properly. So this will happen with enzyme deficiencies. So this can block metabolism at the reactions in the mitochondria and lead to an accumulation of specific substrates causing severe medical conditions. So these enzymes deficiencies could be the enzymes during the beta oxidation or even the carnitine transferase in the carnitine shuttle. This will all inhibit the fatty acid chains from entering the mitochondria. So these disorders can lead to disruptions in the process of beta oxidation, which can cause liver disease or failure, hypoglycemia, fasting intolerances, SIDS, which is sudden infant death syndrome, seizures, or muscle weakness. So lots of things can happen if this process of beta oxidation does not work. But the treatment for this, so these illnesses caused by these disorders are often treated with glucose for immediate cell relief and to prevent suppression of beta oxidation by helping the liver and muscles. So this is just applying a quick amount of energy so our cells can maintain fueled. Because no treatment can lead to death or brain damage. Long-term treatment can include avoiding fasting, so there's no chance for those cells to not get the glucose and energy they need, ATP. Yeah, so that is all for my presentation of beta oxidation. I hope you enjoyed and learned something. Here are my references. Thank you.